Hi friends, I am Arsha Vasudevan of TDMC Alappuzha. Today's topic is apoptosis and intracellular accumulations. First about apoptosis. In this session, we will be discussing what is apoptosis, its causes, morphology, biochemical changes, mechanisms, and some examples. First, about the definition. Apoptosis is a pathway of cell death that is induced by a tightly regulated suicide program in which the cells destined to die activate intrinsic enzymes that degrade the cell's own nuclear DNA and nuclear as well as cytoplasmic proteins. This definition seems to be a little lengthy, isn't it? So, here we have to note the key points from this definition. In the previous classes, we have learned about cell injury. There are two major types, reversible cell injury and irreversible cell injury. What happens if a cell undergoes a reversible cell injury? It may either undergo apoptosis or necrosis. Here we will see what are the features of apoptosis. Apoptosis is a coordinated and internally programmed form of cell death. It means that it is regulated genetically. It's a form of cell suicide. The most characteristic feature to be noted is that it is not accompanied by any kind of inflammation. The reason being that the debris and the dead cell contents are removed before the contents leak out through the plasma membrane. This occurs through phagocytosis. So, no inflammation and no collateral tissue damage occurs. Okay, the key features are it's a coordinated and internally programmed form of cell death, not accompanied by inflammation or collateral tissue damage. Now let's look what are the causes of apoptosis. Causes are physiological as well as pathological. The main reason behind which apoptosis takes place is to remove unwanted, aged and harmful cells from our body. Physiological causes. I'll cite some examples. First one is the development of an embryo into adult. During this process, there are some organs or parts of embryo which regress not found in the adult body. This regression mainly occurs through apoptosis. Next is the involution of cells in hormone dependent tissues after the withdrawal of hormonal support. Examples are endometrial shedding after withdrawal of progesterone and regression of lactating breast. Another example is the normal destruction followed by replacement and proliferation of new cells. In all these cases, we do not see any kind of inflammation. Next, the pathological causes. Imagine a cell whose DNA is damaged by any kind of external agents like for example radiation, chemotherapeutic agents, etc. This cell may undergo apoptosis. Next example is accumulation of misfolded proteins. Normal cells produce normal proteins, but in some cells which are mutated, abnormal proteins may be produced as a result of this mutation, and this may accumulate in the cytoplasm, and this can lead to ER stress. Endoplasmic reticulum will undergo stress, and this triggers apoptosis. Other examples include cell death in certain infections like certain viral infections and the rejection of graft tissue. This is done by cytotoxic T cells. So, The cytotoxic T cells play a major role in apoptosis. So, apoptosis has physiological as well as pathological causes. Next, we look into the morphology of an apoptotic cell. Apoptosis can occur in single cells or in small clusters of cells, but the surrounding cells won't be affected. So the background is that of viable cells. They are normal cells. Then what about an apoptotic cell? Their cell shrinkage is a main feature in apoptosis. We can see round to oval shrunken cells. The organelles in these cells may be shrunken or normal in size. Anyway, the cell size will be reduced. As a result, the same contents are accumulated in a small volume, so the cytoplasm will be intensely eosinophilic. Cytoplasm will be intensely eosinophilic. But in necrosis, what you see is that the cells swell up. But here in apoptosis, the cell shrinks. Next, about the chromatin of an apoptotic cell. Chromatin will be contents peripherally near the nuclear membrane. Next, there will be formation of cytoplasmic blebs and apoptotic bodies. The cells first show swelling, and these swellings finally get detached from the cell to form small parts called apoptotic bodies. The apoptotic bodies has portions of nucleus as well as some amount of cytoplasm in it. The most important point to be noted here it is that the apoptotic body has an intact plasma membrane. The plasma membrane is not damaged so that no contents can leak out from the cell to outside. So the plasma membrane is intact and no contents leak out, and there will be no inflammatory reaction surrounding the apoptotic cell. Further, these apoptotic cells are phagocytosed by macrophages. This is a picture of an apoptotic cell. Here you can see the topmost cell, you can see intensely eosinophilic cytoplasm, and the cell has shrunken. Cell shrinkage is to be noted here, and the chromatin is also condensed. This is also a similar picture. And here you can see the formation of blebs, that is swellings containing cytoplasm as well as portions of nucleus. Then they detach to form apoptotic bodies which are phagocytosed. So what are the biochemical changes taking place in an apoptotic cell? Proteolysis. First one is proteolysis. Proteolysis of cytoskeletal proteins as well as other proteins of the cell occur. Then these lyse proteins form protein-protein cross linkages. Fragmentation of chromatin also occurs by nuclease enzyme. Then, in the outer surface of the plasma membrane, appearance of some molecules like phosphatidylserine and thrombospondin is seen. So the biochemical changes are proteolysis, pyknosis of nucleus, fragmentation of chromatin, and appearance of phosphatidylserine and thrombospondin on the outer surface of the cell. Next, we will move on to the mechanisms of apoptosis. Apoptosis has an initiation phase as well as an execution phase. The execution of apoptosis is mainly carried out by an enzyme known as caspase. C stands for cysteine, ASP stands for aspartic acid, and ASC is given usually at the end of the names of enzymes. Caspase is a proteolytic enzyme. In a normal cell, caspase exists in a spro enzyme or a zymogen, that is in the inactive form. Presence of cleaved caspase in a cell indicates the cell is undergoing apoptosis. Then the question arises, how a caspase is activated? Activation of caspase is regulated by pro and anti-apoptotic genes. Pro and anti-apoptotic genes. Okay. Now let's look what are the two pathways of apoptosis. Intrinsic pathway and extrinsic pathway. Both pathways converge in caspase activation and leads to cell death by apoptosis. First, moving on to intrinsic pathway. This is the major pathway occurring in most of the cases. The main characteristic feature in this pathway is the increased permeability of the outer membrane of the mitochondria. Okay, the most characteristic feature is the increased permeability of the outer membrane of the mitochondria. As the permeability is increased, there will be release of pro-apoptotic molecules like cytochrome C into the cytoplasm. You all will be knowing what is cytochrome C. It is a protein located in the mitochondrial inner membrane which is involved in the ETC and helps in the generation of ATP and thus the survival of the cell. So this molecule cytochrome C escapes out into the cytoplasm. In the cytoplasm, it combines with APAF1, apoptosis activating factor 1, to form apoptosome. So, an apoptosome is formed by the combination of cytochrome C and APAF1. The apoptosome then binds to caspase 9 and this activates it. And caspase 9 further activates another caspases and this cascade goes on. So this is an auto-amplified process and this triggers apoptosis and the death of the cell. Okay. So in the intrinsic pathway, caspase activation is by the leakage of cytochrome C into the cytoplasm. Then the next question is how this mitochondrial permeability is increased, leading to the release of cytochrome C. So this release is mainly mediated by BCL family of proteins. BCL stands for B cell lymphoma. This family of proteins are produced by BCL family of oncogenes. BCL family of oncogenes, when overexpressed, can lead to B cell lymphoma. That is why it gets its name. There are about 20 members in this family of proteins and they are classified into 3 groups based on their nature, that is whether they are pro or anti-apoptotic, and second basis is of the BH domains in the proteins. BH domain means similarity in the domains present in the protein. Domain is a 3D structure of protein. So let's see what are the anti-apoptotic proteins. BCL2, BCLXL and MCL1 are anti-apoptotic. BCL2, BCLXL and MCL1 are anti-apoptotic. They keep the mitochondrial membrane impermeable, prevent the leakage of cytochrome C and thus help to prevent apoptosis. They contain all 4 BH domains, that is BH1 to 4 type of domains are present in them. Next group is pro-apoptotic. Examples are BAX and BAC, BAX and BAK. Their function is to oligomerize in mitochondrial membrane and increase the permeability, thus promoting apoptosis. It has all four domains in its structure. Next group include the sensors. What are sensors? Sensors sense the cellular stress or damage. For example, when there is a withdrawal of growth stimulus or factors, or consider if a cell is exposed to some damaging influence like external radiation. The sensor molecules like BAT, BIM, BIT, NOXA, and PUMA recognize that the cell is under stress. BAT, BIM, BIT, NOXA, PUMA. They further activate BAX and BAC. Thus, they are pro-apoptotic or help in apoptosis. They have BH3 domain only one domain is present in them. So, intrinsic pathway caspases are activated. Caspase 9 is activated, leading to apoptosis. So, main characteristic feature to note here is the increased permeability of outer membrane of mitochondria, and so cytochrome C leaks into the cytoplasm, and this is regulated by pro and anti-apoptotic genes. The pro-apoptotic genes are BAX, BAC, and sensors include BAD, BIM, BIT, NOXA, PUMA, and the anti-apoptotic genes are BCL2, BCLXL, and MCL1. So now we'll move on to extrinsic pathway. Okay. The main feature of extrinsic pathway, or the main factor to note here, is this pathway occurs through the activation of death receptors on plasma membrane. So what are death receptors? Let's have a look on that. Death receptors are members of TNF receptor family. They have a cytoplasmic and membrane bound domains. The cytoplasmic domain is known as the death domain. So that means that they have cytoplasmic as well as membrane bound parts, and the cytoplasmic part is known as death domain. Some examples of this type of death receptors are TNFR1, tumor necrosis factor receptor 1, and FAS. So let's have a look on FAS pathway. The target cell expresses the FAS receptor. FAS receptor is expressed on the target cell. So this figure will show you this is the target cell in the bottom, and this is the white colored thing is the death receptor, the FAS receptor, and the red colored things projecting into the cytoplasm is the death receptor. So you have seen the death receptor and the FAS part of the protein. Then the killer cell, that is the cytotoxic T cell, expresses FAS ligand, that is the receptor for the ligand for FAS receptor. When the cytotoxic T cell comes with its FAS ligand comes and binds to the FAS receptor and on appropriate circumstances, such as when the target cell is affected by some viruses. So the FAS ligand on the cytotoxic T cell and FAS receptor combine. So this leads to the formation of a group. So here we can see 3 FAS receptors are coming together and forming a complex, and together they bind to FAS ligand. And further, the cytoplasmic domain of FAS, thus shown in the red color, combines with the binding site of another protein having a death domain. The last one, the last one you can see here, this combines with the death receptors of FAS to form fast associated death domain, or FADD. Fast associated death domain. This activates caspase 8 and further triggers apoptosis. So this is all about the extrinsic pathway of apoptosis. So in the extrinsic pathway, the extrinsic pathway mainly occurs through the activation of death receptors. I think you have got the concept. What is the mechanism of apoptosis? Further, this after the cell after the cell is destroyed, it undergoes phagocytosis. You have learned earlier that the cell expresses some receptor molecules like phosphatidylserine and thrombospondin. This makes it tasty for phagocytes, so the phagocytes can now easily recognize these cells and destroy them. This picture shows intrinsic pathway. This is a normal viable cell with adequate amount of growth factor. Production of anti-apoptotic proteins is abundant so that cytochrome C does not leak into the cytoplasm. The picture next shows that of an apoptotic cell irradiated. So there occurs DNA damage and the survival signals are lacking. Both these factors contribute in the production of sensor proteins. You already learned that they are BAD, BIM, NOXA, PUMA, etc. So this antagonizes BCL2 and the production of BCL2 is decreased. So BAX and BAD get active. BAX and BAG get activated and leakage of cytochrome C occurs. Activation of caspases which triggers apoptosis. This picture shows both intrinsic and extrinsic pathways at the same time. You can refer to it. Okay, now let's move on to some clinical pathologic correlations of apoptosis. What happens when a cell undergoes defective apoptosis or decreased apoptosis? This increases the survival chance of the cell. So some cells with abnormal mutations like that of TP53 may survive and increase in number. So this leads to oncogenicity. Another problem with defective apoptosis is the increased chance of autoimmune diseases, because normally in our body lymphocytes reacting to self-antigens or self-destructive lymphocytes are destroyed through apoptosis. So the problems arising with defective apoptosis are increased oncogenic potential and increased chance of autoimmune diseases. What happens if apoptosis is increased than normal? Some neurodegenerative diseases are examples of increased apoptosis. Another example is in the case of ischemic injury in MI and stroke. Apoptosis comes into play. Increased apoptosis comes into play. About 80% of the cell death is through apoptosis. And death of some virus infected cells in certain viral infection is also through apoptosis. So this is all about apoptosis. Now we will move on to